So hello guys, welcome to my YouTube channel Med School with Shubham. I am Shubham Mane, a third professional MBBS student, and I shall be teaching you anatomy. I have started a new series on embryology on my YouTube channel in which I have been teaching you all the core concepts of embryology. I have completed the first week of development and also started the second week of development. In the previous lecture we have started the changes in the trophoblast in the second week of development, in which I taught you about the small lacunae are formed inside the syncytial trophoblast, which are filled with the embryotroph, and then in the decidua basalis the maternal blood vessels are formed, and a new connection is formed between the lacunae and the maternal blood vessels, and this connection is called as the uteroplacental circulation. So this is the uteroplacental circulation. These are the lacunae which are initially filled with the embryotroph, and then further it is filled with the maternal blood, and through this syncytial trophoblast, through this lacunae, the syncytial trophoblast will absorb the nutrition, and then it will provide it to the growing embryo. I hope this was very clear to you till the previous lecture. Now we shall start the changes with point of the cytotrophoblast. So let's draw the same thing again. This is the cytotrophoblast. Over the cytotrophoblast we have this This is the syncytial trophoblast, and beneath the cytotrophoblast we have this somatopleuric extraembryonic mesoderm, and above that we have this decidua basalis. I hope I am, yes, okay. Okay, so now till the previous lecture we saw that small lacunae are formed in this syncytial trophoblast, and then maternal blood vessels comes in contact with these lacunae, and now the lacunae are filled with the maternal blood, and through this syncytial trophoblast the nutrition shall be absorbed by the syncytial trophoblast, and now it will go to the growing fetus. Okay, so now what will happen further? So now this cytotrophoblast, it will start proliferating new cells inside the syncytial trophoblast in this way. So they will form small finger-like projections inside this syncytial trophoblast. In this manner, small finger-like projections shall be seen inside the syncytial trophoblast, which are formed from this cytotrophoblast. Okay, these are the extensions of the cytotrophoblast which will be seen inside the syncytial trophoblast. Okay, now what will happen in the next step? This somatopleuric extraembryonic mesoderm shall invade inside this finger-like projections in this way, and now the structure shall be looking in this manner now. In this manner, these are the cells of the cytotrophoblast. Okay, over that we have this syncytial trophoblast. And over that we have the decidua basalis. Okay, let me make a, okay. And beneath this we have this somatopleuric extraembryonic mesoderm. Okay, now what happens? Now you have to answer me one thing. Let me elaborate this thing over here. So what you can see is this is the cytotrophoblast, and this is the cytotrophoblast. This thing is the cytotrophoblastic cells, and this is the somatopleuric extraembryonic mesoderm. So if you have watched my previous lectures, I hope you can very well answer this question, that cytotrophoblast plus somatopleuric extraembryonic mesoderm together forms a structure which is called as the chorion. I hope you remember this thing. Cytotrophoblast and somatopleuric extraembryonic mesoderm together forms a structure which is called as the chorion. And so this entire thing are chorion because they are made up of cytotrophoblast and somatopleuric extraembryonic mesoderm, and they are forming a finger-like projections, and these finger-like projections are generally called as villi, but these villi are made up of chorion. Therefore, we can name them as chorionic villi. These are chorionic villi. Now what will happen? This is also chorionic villi. This is also chorionic villi. And now what will happen further? That inside this chorionic villi, we can see the formation of the new fetal blood vessels. New fetal blood vessels will be formed, and they shall be invading inside this chorionic villi. And this is also chorionic villi. But there are three different names for this chorionic villi. This one, in which only cytotrophoblast is present, those are called as the primary chorionic villi. The one in which only cytotrophoblast and extraembryonic mesoderm is present, somatopleuric extraembryonic mesoderm is present, those are the secondary chorionic villi. And the one in which the blood vessels have been formed, that is the cytotrophoblast, then the somatopleuric extraembryonic mesoderm, and the blood vessels fetal blood vessels are present, those are called as the tertiary chorionic villi. Primary chorionic villi, secondary chorionic villi, and the tertiary chorionic villi. See, this chorionic villi formation is itself an important short note or a 6-marker question in the various university exams. So another important question that comes in the exam is the fate of the chorionic villi. So now we will be learning about the fate of the chorionic villi. Before learning the fate of the chorionic villi, let me tell you an important thing. So I hope you remember this thing we saw in the first lecture of the second part of the first lecture of the second week of development. In this we saw that this were the epiblast, this is the hypoblast, the amniogenic cells, the Usher's membrane, the amniotic cavity, and the secondary yolk sac. Initially it was primary yolk sac, then it became the secondary yolk sac. This is the this everything is the extraembryonic mesoderm. Then we got the formation of the extraembryonic coelom. Because of the formation of extraembryonic coelom, the extraembryonic mesoderm further got differentiated into somatopleuric extraembryonic mesoderm, which is in contact with the cytotrophoblast and the amniogenic cell, and the splanchnopluric extraembryonic mesoderm, which is in contact with the yolk sac and the Usher's membrane. I hope this is very clear to you guys now. And the place where the extraembryonic coelom is absent, is called as the undifferentiated extraembryonic mesoderm. That part of extraembryonic mesoderm where the extraembryonic coelom is absent is called as the undifferentiated extraembryonic mesoderm. Now, you might be thinking that the undifferentiated extraembryonic mesoderm is the one which is present above the amniotic cavity and the amniogenic cell. Because I have shown you in this diagram that it is above the amniotic cavity or the amniogenic cell, but it is not like that. What will happen now? I will turn this image in this way. I will turn this structure by 90 degree, and now you can see something like this. So what is this? The actual undifferentiated extraembryonic mesoderm is the one which is attached to this bilaminar germ layer, and then from backside it will move upward and backward. It will move backward and upward. It is first moving backward and then it will moving upward. So it will move backward and upward. So whenever you see the diagram from this side, you view this diagram, so you can see that this part of the undifferentiated extraembryonic mesoderm is something which is above to this amniotic cavity. But it is not like that, because in this you can see that it is above to this amniotic cavity, but in this image you can clearly visualize that it is moving from backward to the upward. Therefore, when you visualize from this side, you can see that it is something which is above to the amniotic cavity, but it is going from backward to the upward side. So for that I shall be showing you a demonstration. Assume that this is the amniogenic cell and this is the amniotic cavity, and this is the Users membrane, and inside that is the primary or the secondary yolk cell, and this is the bilaminar germ layer. Now assume that this is the undifferentiated extraembryonic mesoderm. I have been attaching over here. It is moving from backward to the upward. So when you visualize from this side, you can see that it is moving slightly upward. But when I change the view, now you can see that it is something which is above to the amniotic cavity, this side. Now when I change the view like this, you can see it is in this way. But when I show it in this way, you can see that it is above to the amniotic cavity. But it is actually going from backward to the upward side. I hope this is very clear. Now let's come to the fate of the chorionic villi. So the primary and the secondary chorionic villi will be formed all over. Means this is the primary chorionic villi, and further this primary chorionic villi will be different, will further progress to form the secondary chorionic villi. Okay, the primary and secondary chorionic villi will be formed all over. All over it will be formed, but the tertiary chorionic villi or the blood vessels, fetal blood vessels in this secondary chorionic villi will be formed only in those chorionic villi which are in contact with the undifferentiated extraembryonic mesoderm. Means what? So these are the, assume that these are the secondary chorionic villi. Okay, but the fetal blood vessels will be formed only in those secondary chorionic villi will be present in contact with this undifferentiated extraembryonic mesoderm. Means only in this part, because these are the secondary chorionic villi which are in contact with the undifferentiated extraembryonic mesoderm, and through this undifferentiated extraembryonic mesoderm the nutrition shall be reaching the actual growing embryo. Now why is this important? Because if the blood vessels are formed here, if the blood vessels are formed, assume that the blood vessel is formed in this chorionic villi. So the lacuna will be present through which the blood vessels, through which the nutrition shall be absorbed, and now this chorionic villi will now absorb the nutrition and it will take inside. It will reach the somatopleuric extraembryonic mesoderm, and from somatopleuric extraembryonic mesoderm it will reach the coelom. And now most of the part will be reaching the coelom and only some part will reach this growing embryo, which we don't want to happen. We want that the entire nutrition should reach the embryo, growing embryo, and the required part should enter the coelom or the somatopleuric extraembryonic mesoderm. So for that we need a direct connection between the chorionic villi, or between the source and the, this is the source of nutrition and this is the utility of the nutrition. So this is the direct entry, this is the way or the direct entry towards the growing embryo. Therefore the blood vessels will be formed only in this part of the chorionic villi which is in contact with the undifferentiated extraembryonic mesoderm, and rest all the chorionic villi will get shed off because now they are of no use. So all these are called as the, they are going to leave now, therefore they are called as the chorionic levae, and these where the tertiary chorionic villi will be formed, they will form a tuft of chorionic villi over there, a bunch of chorionic villi will be formed, and these are called as the chorionic frondosome. The chorionic frondosome will be the structure that will be forming the placenta, and this is the undifferentiated extraembryonic mesoderm. It will be further forming the connecting stalk because it is connecting the growing embryo with the placenta. So it will form the connecting stalk, and this connecting stalk will further form the umbilical cord. I hope this is very clear to you guys. This was all about the development of the chorionic villi and the fate of the chorionic villi. The development and the fate of the chorionic villi formation is itself a very important question in the university exam, and also it forms the base for the placenta formation lecture. I hope this is very clear to you guys. Let me revise everything that we studied from the part 3 and the part 4, because both of them are in continuation with each other. So we started in the previous lecture, we started the changes in the trophoblastic layer of the blastocyst. So blastocyst further got differentiated into trophoblast and the embryoblast. Trophoblast got further differentiated into cytotrophoblast and the syncytotrophoblast, the cytotrophoblast and the syncytotrophoblast, and the embryoblast further got differentiated into epiblast and the hypoblast. After implantation, this defect of the implantation shall be sealed off. This is the defect of implantation, or this is the remaining part which is opened, through which the opening through which the blastocyst has invaded inside the uterine cavity. It has been plugged off, or it has been sealed off with the fibrin plug, which will be further covered by the decidua capsularis, and this is the decidua basalis. Okay, so this is the let me tell you one more thing. This is the syncytotrophoblast that is growing more towards the embryonic end. This is the embryonic end, this is the abembryonic end. So it will be growing more towards the embryonic end. Now let me elaborate this part over here. So this is the cytotrophoblast, this is the somatopleuric extraembryonic mesoderm, and this is the syncytotrophoblast, and this is the decidua basalis. Inside the syncytotrophoblast, first we will see the formation of the lacunae. These lacunae will be initially filled with the embryotroph, which is rich in glycogen. Then in the decidua basalis, we can see the formation of the maternal blood vessels. These maternal blood vessels will now come in contact with this lacunae, and now a new circulation will take place, which is called as the uteroplacental circulation. Now we saw the changes in the cytotrophoblast also. Cytotrophoblast starts proliferating itself. It will form finger-like projections in the syncytotrophoblast. Now these finger-like projections, now this somatopleuric extraembryonic mesoderm will invade inside this finger-like projection, and now it will seem to be in this manner. This looks like villi, but which are formed up of chorion. Therefore, they are called as the chorionic villi. Also, now the blood vessels and fetal blood vessels are formed in this chorionic villi. Therefore, now we can divide them into primary chorionic villi, in which we have only the cytotrophoblast, the secondary chorionic villi, in which we have the cytotrophoblast and the somatopleuric extraembryonic mesoderm, and we have the tertiary chorionic villi, in which we have the cytotrophoblast, somatopleuric extraembryonic mesoderm, and the fetal blood vessels. Now let's learn about the fate of the chorionic villi. So the primary and the secondary chorionic villi will be formed all over the embryo, but the tertiary chorionic villi will be formed only in that part of the embryo, only in that part which is in contact with the undifferentiated extraembryonic mesoderm. And now this primary and secondary chorionic villi, which is formed all over, they are going to leave this embryo. Therefore, they are called as chorionic levae, and the tertiary will be formed only in those chorionic villi, or the blood vessels or fetal blood vessels will be formed only in those chorionic villi which is in contact with the undifferentiated extraembryonic mesoderm. So those are going to form the placenta. It will stay with the embryo till the birth. So they are called as the chorionic frondosome, through which the placenta will be formed, and this part is the undifferentiated extraembryonic mesoderm. This is the only part where the coelom is absent. This is the coelom which is surrounded all over, and this is the part only where the coelom is absent. So it forms a direct entry into the growing embryo. Therefore, it will form the connecting stalk and the umbilical cord. I hope this was very clear to you guys. Please like my videos, share my videos, and also subscribe to my YouTube channel. Thank you so much, guys.